University St. Louis School of Medicine. Throughout his career, he leveraged his vast experience and understanding of the fundamental principles of retina function to evaluate the mechanisms at the cellular level of neurodegenerative diseases. Dr. Sagdullaev and his team at the Burke Neurological Institute explore innovative scientific approaches to vision repair in animal models that can be translated to humans. Botir's work has resulted in numerous high-profile, peer-reviewed publications and lectures around the world. His expertise in physiology and imaging has earned him continuous NIH funding and strategic collaborative partnerships and awards. In 2008, Dr. Sagdullaev left Columbia University to join the team at the Burke Neurological Institute. He is an associate director at the Institute and an associate professor of ophthalmology and neuroscience at Weill Cornell Medicine. Botir is also the director of Laboratory for Visual Plasticity and Repair at BNI. Botir, thank you for taking the time to teach us today about your groundbreaking work.
All right, thank you so much for inviting me and everyone for joining this webinar. And I'm going to start sharing my screen and we'll get going about the presentation. So now everybody, you guys can see it now? All right, so it should be on your screens, right?
Yes, all set.
All right, so as Christine suggested, that the goal of today's webinar is to illustrate you our study of brain and how we can use this knowledge for developing repair strategies to treat common neurological disease. But the main goal also is that to keep everyone along the talk, and for that, this title, we would like to begin with the title. The title is 3 lines long. It's very descriptive, but I like a more revised title offered by Yogi Berra that basically indicates us that we can learn a lot about the brain function of anything by simply watching. And attentive watching is a very important aspect also in the understanding how brain functions. And this is my revised title of the talk for today. And where do we begin? If we start looking at the history, the important part of learning is to know where to look and what tools you use to look. And throughout the history, if you look here, you can go almost 3,000 years ago with this primitive lens to amplify the images that people were wondering about. What are they looking at? And then up to today, all these giant imaging devices in medical facilities or research facilities, and everything between different sides of microscope. And as you see here, we're going through 3,000 years ago to the modern times, and yet the most advanced and oldest imaging device that nature ever created we have it, it's eye. We have 2 of it, hopefully, and many animals have it, and it is old, and in this particular shape, more or less recognizable shape, it is about 500 million years old, starting with jawless fish and mollusks swimming in Silurian ocean many, many, many years ago. The important part of it is that we all have it. And another aspect is it's a model system to study the brain, because this is the only approachable part of the brain. So next time you look into someone's eyes, just keep in mind that you're looking not in their soul, but you're looking in their brain, because through the pupil, you're looking at the retina, and retina is a part of the central nervous system in the brain. So another important part of the visual system in the eye particularly is that it has a natural stimulus, light. And importance of the system cannot be underestimated, because 84% of our interactions with the world around us relies on visual input, and visual impairment is one of the most leading reasons for disability and impairment. It's also, as a part of CNS, contains all essential blocks, and this is I will walk you through during my talk today. And it also very important is that it is a site of common disease here. And do you see my cursor here? I'm pointing in front.
Yes.
So good. So I will describe the structure of the eye. I'm going to spend a little time on this slide because I want to bring everyone along why it is important in terms of structure, function, and also its relationship to common disease. Many of you might have it. If you don't have it personally, somebody you know close will have it. So front of the eye is an imaging device. Obviously, it's an optic system. Its ability, it's a need to focus the image on the back of the eye, which is the retina, which is basically imaging sensor. In the old cameras, it used to be film. Now it's a microchip with a detector, light detectors. But eye structurally, it's very functionally, it's a very similar system. So to do that, it has a set of lenses. There is one lens which is cornea, and the second lens, it's a lens called crystalline or lens. So if something happens to them, if you wear glasses or corrective lenses, that means there is something going on with the refractive properties of these two lenses. If you have a cataract, they're no longer transparent. If we move further down the path of light, there is a vitreous, which is fills up all this inner volume and keeps eye pumped, because you need to have a very rigid structure of the eye to keep the axial length constant here. So as people age, usually if you have fluorescence in the eye, it means the substances, proteins within the vitreous aggregate together, creating these large non-transparent elements that appear as a fluorescence. If you age longer, you might have started seeing a lot of the bigger fluorescence. That's usually when vitreous becomes detached from the retina, because it's attached here strongly, but with the age, when it dries out or eye gets longer, it detaches. That's causing lots of fluorescence. Sometimes it might lead also to the detachment of entire retina. This is a retinal-specific disease. Now, if we move further to the nervous part of the eye, the retina itself, that's where you become striking some disease that are maybe common to the entire brain system and the body, like diabetic retinopathies, all neuropathies that affect neurons and other cells in our brain, and also a disease like stroke that have relation to the blood circulation. For instance, if there is a blood clot collected in the periphery, makes its way to the fine capillaries within the retina and clots the one of these capillaries, it might cause a retinal stroke, and there is such a thing. It's quite common thing. And many diseases such as Alzheimer's and Parkinson's have their imprint on the visual system as well. But today I will, at some point in a few slides, I will start talking in the general disease, metabolic disease such as diabetes. And the interesting thing is diabetes has a very major implication to brain and to the visual system in particular. And but where do you begin? Well, obviously, you can start by simply looking into the eye. And this is not human eye. As you see, there's too many hairs here. This is a mouse eye. And human eye and mouse eyes share 99% commonality. There are with few distinct differences, which I will point down later. You can actually take live mouse and look into eye, and you can start simple imaging device. You can start looking through the eye. And it's like in humans, when you go to optometrists, they look into your bottom of your eye. It's a fundoscopy called. You can see all these nice blood vessels and the part of the retinal system. If you use more sophisticated device such as the confocal microscope, you can actually pierce through different layers of retina. Retina is like a sandwich. It's like any part of the brain. It has multiple layers, and each layer represents a different structure that carries different function in the brain, and retina is no exception. You can move further. You can do this something like this in terms of video, not just still images. And when you do that, you can, in a large area, you can start seeing the frame shift. It's actually animal heartbeat, and that's why it's a motion. And if you look at all this flickering inside, this is actually blood cells moving through the blood capillaries within the retina. And this is all in live animals. And when optometrists look in your eye, sometimes they can even detect this. And that's what I mean by the only accessible part of the brain. You don't need to do much to actually visualize how things evolve and what's happening in the brain as you go. You can use a more sophisticated tool, like a 2-photon imaging device, where you can really zoom in into a small area. And by doing that, you can actually see how these individual blood cells flow through the brain. And this is how you can see by naked eye, augmented by some advanced technology. Well, if you start applying different tools, you start labeling different types of cells within the brain, you can start appreciate the full beauty of the system. And here, this is dissected retina, a label for certain elements within it. And you can recognize that capillaries, all these blood vessels, they feed the tissue. These green cells, this is a cell bodies of neurons. Those are smart cells. And this is glial cells. Their primary role up to recently was considered to be a supportive role. They nurture, they provide support, structural and functional support to everything around it. And these three are the building blocks of the brain, retina and any parts of the brain. And you can see how tightly they interact together. But despite of this elegant design and beauty, brain has one major flaw. And its major flaw is that it lacks energy backup. So any part of the brain, the energy is stored. For instance, if you tie up your limb or leg or arm for 10 minutes, it gets sleepy, but eventually recovers its function, and it's okay. It's because there is a lot of energy stored in the system. Our whole body has a fat as a store system, but brain fat is not a storage device. Brain's fat is an active ingredient. It's a primary element. That's neurons are made of largely fat, and axons made up largely of fat. And if you, for a few minutes, restrict the blood flow into the brain, it's usually that's a major cause of stroke disease, ischemic stroke. And if you more than, let's say, 5 minutes, it's irreversible damage and death. So, and that's why if you don't believe these facts, then you don't need to go further. You can even it made its way on one of these Snapple caps. Here I have here. I went with kids to a pizza joint, and we had a Snapple once a week. They allowed to have it, and you have one of those. And it says that human brain takes up 2% body weight, but consumes 20% energy. It is one of the most energy demanding parts of the brain, part of the body, and the retina is on the top of the list within the brain itself. But before you decide to teach your kids or homeschool your kids of the Snapple caps, there is a warning that some of them are less credible. For instance, one of these, which is totally untrue and cannot be trusted. So there is a word of caution on this. So now, when you look into the eye again, and we just revisit this briefly, it's at high magnification, the same view. You can see all these, this is smart cells, neurons, there is a capillaries, those are red blood cells, those are red blood cells floating around, this is the neurons, and this is the capillaries again. And you're gonna see this, neurons, one of the cells that I was looking at before. And around each cell, you have very dense vascular network, because for these guys to do their job and to live, to be alive, they need constantly be nurtured and supplied by the oxygen, glucose, and also not only deliver good stuff, but also remove the bad stuff and clear up the trash from the circuits in the brain. And if you look at the eye as a whole, what we're looking here is a open up retina of a mouse with its entire vasculature. So there's a big vessel, this is a vein, this is an artery, and everything fine in between. Those are capillaries. And the green cells, this is randomly labeled neurons to illustrate the scale of the vascularization, and how, because when the system needs energy, there is a logistics that must deliver it, and this is beautifully illustrated in this slide. This is mouse retina. Human retina would look very, very similar, with one exception. On this spot, you will have us as humans will have no vascularization. This is a fovea. This is area of fine vision, because imagine light going through the system. It has to go past all these structural things, and somehow we don't see them because brain visual cortex makes up for this and tends to ignore this. And the fovea, to increase that for fine vision, fovea doesn't have vasculature. It's only in the peripheral supply, and everything else is supplied by diffusion. So now it's not only blood supply per se. It's on demand, because when it's being used, that's when most energy is needed. When it's not used, you don't want to waste your energy, so you need to ramp down the blood flow. And this is one of the key tools for diagnosis of that's used in diagnosis. This is a blood oxygen level derived MRI, magnetic resonance imaging. This is a very common tool where usually it's a brain scan for diagnostic power. And what you actually, it's inferred to indicate the brain activity. In fact, actually, it doesn't indicate brain activity. It indicates oxygen exchange and the blood flow. And it's inferred to brain activity because if there is more brain activity, it's going to trigger more blood flow. And for this, you need it's not only just fine system. This capillary network, it's also have a three-dimensional structure. What I showed it was kind of flat, but if you go layer by layer, as I said, a sandwich, you will find it has this beautiful layered structure. And there are sites where the certain areas, it's like bottlenecks, regulatory sites, where the blood vessels interact with the neuronal input, with the activity sensors. And that's where the neurons, the smart cells, tell blood vessels that, hey, now we are working hard, and this is time where we need more oxygen, we need to get more blood flowing to support this function. And once the signals ramps down, blood supplies reduces. And this is, let's say, in one of those capillary layers within the retina where this exchange takes place. And the red is color-coded. It's basically blood vessels. And the green, you see how much neuronal inputs they receive. They talk to each other a lot, and they talk to each other constantly, and their conversations change with the activity of each component. And our goal in general is to understand the cellular interactions that mediate this. And we know, because these interactions are essential for normal brain function. And these interactions are usually the first things that alter at the onset of many neurological disease. Not just neurological, any disease in the body, but because brain is so much reliant on uninterrupted blood supply, this becomes a very early indicator of things that go wrong. And I believe this is the point where we need to pay attention the most, because sometimes when the manifest changes take place, it might be too late to treat it. And one of the aspects of the whole thing is to look at this and understand that this is not just made to look beautiful. It's not just a form. It is a function of the brain, and it just happened to be look so spectacular. The tragic irony of this cap that I have in my hands is that it's written on the soda can, the main source of pandemic across the world, particularly in the United States, cause of diabetes, because sugar drinks, it's like Steve Jobs used to tell John Scully, are you going to sell sugar water, or are you going to join and change the world, join Apple and change the world? So he ended up changing Apple, but rest of history. But back to the talk is that diabetes is not just metabolic disease. It's not only sugar exchange disruption in the body. It's actually one of the most common cause of neurological impairment. For instance, in the retina, if you didn't know about it, it's that 45% of people who have diabetes at some point will develop visual impairment and vision loss. It's a very dramatic. And in terms of sheer number, there are tens of millions of Americans that are affected by this. In fact, it's more prevalent than glaucoma and age-related macular degeneration combined. And this is a lifestyle disease. There is something we can do about it, and yet we're doing very little about it. And that's why, hence, we need to understand about things that we can change eventually. So what's the hallmarks? When do we need to intervene? Obviously, change your lifestyle, if not an option, it cannot be done. Then we need to look early in disease, because early impairments in the blood flow, and then later it's a disruption of anatomical change like blood retinal barrier. That's basically integrity. Blood cannot leave the blood vessels. If blood leaves blood vessels and spills out into the brain, it is toxic, it is fatal. It is a cause for hemorrhagic stroke. And the similar can happen in any tissue, that a brain tissue that is exposed to the to have a very high blood circulation, and retina, as I mentioned, one of those. But what do we target? Where do we begin, and what's the best things to leverage? Well, for that, let's look at the history of disease. If you, it's in the mouse. If you go through, let's say, through the time points in the mouse progression of diabetes that is induced by certain agents injection that kills cells that produce insulin, it's going to go through a number of events. You don't need to understand many of it. All you need to understand that this is a evolving nature. It's not like onset overnight. There are things pile up over time. In a mouse, it's say months. In humans, usually this will translate to, let's say, 10, 20 years. So the window to act is wide. There are many opportunities to change things. If you look at those, what you notice is that lots of, we diagnose this disease usually based on the appearance. It's a structural. When they look in the eye, like I showed you before, using fundoscopy or fluorescence angiography, they look for anatomical changes. But the early changes usually functional. It's a reduced capillary blood flow, early changes in the ability to see, and those sort of things. This is why there's a key things that we need to keep in mind. Early detection is a key, because you want to prevent this domino effect of things piling up and going past the point of non-return. And the second is that to get there, we need to understand the fundamental basic biology that drives the whole system in control in a healthy situation, and how these things change when disease strikes. And where do we look? Well, we look at the structure. This is kind of, I made this cartoon a few years ago that illustrates the architecture of the retina. But it's also illustrates architecture of entire brain, even the spinal cord. It's the same essential element. Some cells might be different, but essentially you have a set of neurons that talk to each other, and they stimulate and interact with the blood vessels. So, as I said, one of the first elements that goes bad in many diseases, and particularly in diabetic retinopathy, is a reduced capillary blood flow, reduced perfusion of the vessel. So inability to supply the tissue with needed oxygen and glucose and other regulatory factors. So the question is here, then what does regulate this blood flow in these structures? So if you look, there is basically large vessels, artery. This is how blood comes in. Then it flows through arterioles, smaller arteries. Then it's capillaries. Those are fine arteries where you have individual cells passing by. And then it's taken away through the collection of the venules, then veins, and taken back to the heart, which goes to the lungs and enriches with oxygen and brings it back here. And this circle goes on and on and on. So at the capillary layers, where 84% of action takes place, and this is the only side of actual metabolic exchange with the tissue, what does regulate that blood flow? And the blood flow is basically liquid blood flowing through a set of pipes. And what regulates the flow of the pipe? It's basically diameter of your hose. So basically, to regulate the blood, one of the things is to change the diameter. And those are cells that regulate it are these green cells here called pericytes. It's very highly specialized cells in these structures that that control the diameter of these vessels. And there are also smooth muscle cells on the arteries that also change the influx of in the large vessels. Well, let me tell you a little bit more about what these parasites are. Parasites, it's very specialized cell. They're receiving a lot of attention recently because of their numerous functions. Some of them are listed here. It's a change in diameter of the vessels, and by doing so, changing the rate of blood flow and blood supply. They're also important for this separation between blood and the brain tissue itself. That's a blood-brain barrier term. Vascularization is basically how much blood vessels supply the area of the brain. There has to be a specific level of, it can be too little, but it can be too much. And the parasites are shown to control that ratio between tissue and amount of the vascularization of it. Inflammation, if there is something goes, how brain reacts to the insults, and the parasites are usually intermediate of that response too. If response goes over the board, it's going to cause inflammation and actually severely impair the brain function. They also have stem cell properties. What is a stem cell properties? What's its ability to generate new cells. You'll be told over years that, oh, brain cells don't regenerate. Well, newsflash, they actually do, and some of them do more than others. And there are some regions they do more than they do, and some regions they don't. And the parasites in one of those cells actually can regenerate. So one envisions using them, leverage them to actually change their fate and repopulate brain cells that are lost. That's another venue to study. But when we'll just look into simply the circulation of blood in the brain and the retina in particular, this is how it's structured. So on the left side, I made this hybrid image. You have this vasculature. On the right side, all these muscle cells, parasites and smooth muscle cells that control their diameter. And you can see they kind of create this scaffolding or skeleton around each blood vessels and densely populate all of them. And here it's a little bit high zoom. You can see how it nicely populate the walls of the blood vessels of different caliber. You see there is a branch, arterial branch coming in, and this is a vein branch taken out, and this is all capillary, and all of them have one or the other sort of cell, like parasite, that control their diameter. And actually, phenotypically, it's very easy to tell a difference. You would be wondering, how do I tell which is artery and which is vein? Well, artery looks more like oak tree. You see the branching pattern. There is a big stem, there is a smaller branch, there is the smaller branch, and it's the oak tree. And if you look at this, it looks more like pine tree. And why it is that way? Because if you look, once you take a walk today, just pay attention to the difference between oak and a pine. Pine might have small branches coming right out of the major trunk. And this is the same way. Capillaries can go straight into the vein, and usually rarely go straight into the artery. And that's how the phenotypic appearance is very different between these two. But to the point is that the appearance of all these cells changes with the placement, their placement on the tree branch, and which coordinates with the blood flow. If blood comes there, as blood flows, the elements that control blood flow change in their appearance. And this is, here we use some genetic trick to sparsely label them. So not all of them are labeled, one here, one there, and it gives you kind of sense of how they look individually when they're not non-overlapping, when there is no such a big mess. So again, this is artery. In artery, this is smooth muscle cells that are very strong. They express a lot of muscle proteins. They can constrict and dilate very easy in a big way, but by doing that, they're gonna change the blood in a large area. In the brain, usually it's a very site-specific. For that, you need a fine-tuning, and that fine-tuning accomplished by second layer of cells, this cell called sphincter cell. It's kind of faucet that opens and closes blood flow in a very local region. If this cell is activated, blood is permitted to flow here. Here it faces different cell that is more like this, but it's kind of not quite like this. And then you have these cells that have a less of these fingers that control blood flow, but they do it in a much fine way. And here, when you separate them, this is again, it's an artery, blood flows this way, then it goes through sphincter cell, then it goes to precapillary cell, then it goes to parasite, and ability to fine-tune changes from really coarse modulation to letting in or no to much finer control of blood flow, and this is very important. So as I said earlier, that capillary blood flow, these dynamics are altered in many diseases early on, and particularly in diabetes. And in this case, diabetes is just a model system. You can take any disease and you will find very similar chain of events. Then where do we look? And well, one way of scientific study is to advance a hypothesis. And the beauty of hypothesis, it makes a proposal. We say, hey, things happen this way, and if they do, then we confirm hypothesis. If they don't, we reject hypothesis. So the key feature of hypothesis is to be rejectable, because then rejected hypothesis is equally valuable. It means what things not happening, there must be something else is happening. So in our study, we decided, hey, if you separate this vasculature or the way blood supply, the same thing happening in heart, on other parts of the brain too, there might be several things that happen early in diabetes. Either those cells that's supposed to dilate or constrict the capillaries to let more blood in, no longer capable of doing this, or communication with them is disrupted. And these are two hypotheses that I want you to walk through for the rest of the talk and see how we actually peer into the brain function to understand how it works. And that builds our case on where to actually act. Once we identify which component is altered, maybe all of them above, as I show you, then we can target them for repair. So this is our kind of for today's couple of things. So now, I want to add that, as I showed you, there is a lot of diverse cells that control blood flow. We can actually genetically and acutely, we can identify them if we wanted to. And this is important because then we can target them for manipulation in this way. And this is illustration when there are some cells identified that are capillary cells. Here there is a cell that is in a larger vessel. So we can also, and the important part is that, are we going to manipulate these guys, or we're going to manipulate these ones? And gives you more nuanced approach, because you don't want to change everywhere. You want to change where it is affected, because the main reason is not only cure, but do no harm. So, and that's why we need to have this nuanced approach and being able to distinguish one from another. And if you look, they also populate different levels of the building, of the brain building. And one of them one layer, another one another layer, another layer. And this structural arrangement has very important function implications, as you might imagine. So now, the question is, how do we study this in a more nuanced way? As I said, we need to be able to distinguish one cell action from a system action. And that's, with that precision, will let us to create something that is more specific and less invasive. And one way to do this is to basically, like electrical engineers would do, would just take a piece of brain and actually start going around with electrodes and measuring activity of individual thing and finding out which one is altered. If you have something broken, like AC broken, the guy is not idea is not to replace the entire system, but find is the capacitor altered or you need more charge. And for that, they do diagnostic. And that's a very similar approach that we're taking here. So what you're looking here, you're looking at the piece of retina in the dish. And this is that capillary branch. And here you see that electrode where we have very fine system where you can actually activate or manipulate individual cells. And to outline the basic outlines, the capillary and those individual pericyte cells, those cells that control the diameter. And I can actually go there and change the, stimulate one but not the other and see how things change. So remember, hypothesis that one was that these guys no longer active. So how to test it? Well, let's stimulate it and see whether it's going to respond or not. So, and this is what I actually did here. So I'm going to just basically zap this cell and see what happens. And this is a healthy tissue. When I zapped it, you can see things start moving. This starts squeezing, and I zapped it in a way so it's actually constricted. The same way you can zap, so it dilates it. It's easier to see constriction. That's why I'm showing this. And there are two things happen here. First, it was a local response, and then it propagated to involve, to activate other guys. It's basically chain reaction between the members residing on the same tree. This guy apparently talks to other guys and upstream and downstream. So now this is basically before, and this is how it happened after. That's the kind of interesting element. So now let's look at early stages of diabetes. So we have a very similar looking structure here. There is capillaries, arterioles, and this and that. And I have electrode, and I'm stimulating the same cell. And this is before any cell loss. This is very early. There is no vision decline, there is no structural change, but the animal has been diabetic, and we detect early capillary blood flow reduction. So I'm going to do the same experiment here. I'm going to zap this guy and see what happens. So here. So it's kind of hesitant. And then it does react. But you can see it's different than was in the healthy tissue, non-diabetic tissue. And it's different in the way it somewhat responds here, but it does not talk to its neighbors anymore. And as you know, the blood supply along the network, it's a network interaction. It's not one cell function. So if activity change here, this signal has to go upstream to tell, hey, I need more blood. If it doesn't talk to them, there will be no more supply. The logistic chain is disrupted in this case. So, and this is before and this is after. And we can quantify in many ways. Here, it's by amplitude of the diameter change and also spread of it. And that's what gives you profile here and here. And you can see kind of, if you do more of this, you will appreciate that it's not only strength of this individual cell altered, but also most important is how they communicate. So there is a loss signaling between these cells. So it's not as much as ability of one cell to react to the activity, underlying activity. It's failure to communicate to the supplying branch to bring blood flow in. The question now is that, well, how these cells communicate with each other. So that's another fundamental biology question that potentially has a implication to treatment. Yeah. Well, they communicate is a very interesting way. It's like a set of electrical wires. They communicate by electricity. And that electricity travels from one cell to another through a set of pores. These are basically holes in the membranes of adjacent cells, two adjacent cells that have pores that are opposing each other. And that's how they talk. This is a faster communication in the body. It's not chemical synapse. That's why it's called electrical synapse. Basic ions and other molecules can travel in that and signal. That's how one cell will talk to another. And that's the fastest way to do it. So, and in the retina or many vascular cells, this is a particular type of protein. It's called connexin 43. It's a connexin that's a protein that forms these pores between the adjacent membrane. So we have antibody. There is a way to basically have antibody and that selectively attaches to them and that has a color tag to it. We can actually visualize and label them. And here that's labeled in this image here, but just highlighted here. You can see a lot of this protein, and particularly these stringy things and punctuated of these dots, a lot of within the blood vessels and around. So if it's a non-diabetic tissue, if you take a diabetic tissue, you will see there is still some, but there's something fundamentally different between this left and right. And what is different is the selective loss of these string-like structures. We name them vascular relay. These are connections between vascular cells that mediate that fast interaction from the periphery through the capillary to the feeding artery. That's why it's hence named relay. They one to another, the signal jumps and tells, hey, the activity took place, open up here, and we need more blood here, but not there, just here. And that's how it's very selective. And this is a loss here, and this is just summarized. You can ignore it. The point is that to what extent and which cells talk to each other? Is it parasites or it involves other players, or who is mediating this? Because we need to know who we want to target, who we want to treat. The only ones that are malfunctioning, obviously. Well, and there is a very interesting way to do this. Basically, it's kind of a social network of the vascular cells here. And if you want to if you want to see who's talking to who, you just give something to one and see who this cell going to share with. And this is called a neuronal tracing. In the same way, you spread some rumor and see if they talk to each other, this guy's going to talk about the same thing, but not the other ones. So that's how you determine, hey, these guys actually directly talk to each other. And we can do this the same thing in the brain. We can inject a dye that is small enough to penetrate through those pores and diffuse into neighboring cells. And that's tracing. And when you do this to the retinal system, you inject this cell and you wait 15 minutes. And this dye spreads. And it identifies cells that are in direct partners to which they talk to each other, and meaning where the signal spreads in the brain system. It's in the retina, it's example, but obviously it happens in other places too. And this is beautiful, and it's telling us that, hey, there's something very interesting to this. They're very selective between each other. Keep in mind that this is not just isolated vasculature. This is a retina brain tissue. It has thousands of cells around it, and all of them have some sort of communication devices. And yet, when you inject a vascular cell, the signal stays within the vascular cell. So somehow they know each other, and they distinguish each other, and they exclude others from their conversation. And if you do the same experiment to other cells along the vascular branch, or outside of the vascular, say, glial cells, those support cells, you will find out, looks like those cells are very segregated. Like cells want to talk to like cells, but not to other. And neurons like to talk to neurons, glia like to talk to glia. Vascular cells only primarily talk to vascular cells. And things might get a little bit complicated, but I hope you still can follow me. If you look at all these cells, it can create you sort of social network of the brain. So here are the cells, different types of cells that can be injected with this tracer. And down the road, those are the cells that are talked to. And the color indicates the amount of chatter between them. So the dark red, it means a lot of spread. So basically, parasites talk to parasites, and also endothelial. So those are vascular cells that are on this branch. But they don't talk to smooth muscle, so the signal doesn't go much here. Remember that sphincter cell here? Probably it prevents stopping from going. And why do you need this? Well, you need that for a reason that you want to be very local, site-specific response. Remember, there is one way to wash. There are several ways to wash hands. You can open the sink in the bathroom and wash, or you can open the valve entire house. It's going to flood everywhere, but you will be able to wash your hands. But this is very non-specific and energy wasting. You want to open only in one place where you want to open, and lack of the spread guarantees that that actually happens. So now, if you block gap junctions or that connects in or in diabetes, it's just they no longer talk. This one cell picks it up and it goes just cold. It doesn't socialize anymore. And another interesting part is that's again, it's a basic illustrating you from different things. So let me walk you through. So interesting part is that this is healthy tissue. You see, when you inject this cell, it maps how they talk to each other. It's in the reverse into the blood flow. So they signal here and the blood will be supplied. If you block gap junction, it doesn't happen. In diabetic retina, it's you don't see signal travel. You don't even see the branch. There is a big branch here because it's not highlighted. You won't even see it. Pre-diabetic, very important condition, because any sugar level above 250 or below 250, like 200, 250, considered to pre-diabetic. Even that case, when there is no diabetes yet, you already see changing. It's a lot of coupling, right? It is. If you look at the map itself, but if you look how it's happening, it's very different from here. Here, it's somewhat directional. It happens primarily in the direction of blood supply. Here, it's erratic. It's all over the place and not necessarily in the direction. That's directionality is very important part of the whole interaction. And this is kind of illustrated here, that in this type of disease, you have a lost sense of the directionality. So you want to signal where the blood comes from, not where it's going away. And in diabetes, it's like there is no signal at all. Early stages, it's already skewed and it's losing that important feature. Well, and here we come to the almost end, is that in a summary that there is it's a kind of scientific term, summary, but what I want to convey here, there is a very interesting cell-to-cell interactions that happen in the brain. They happen in a healthy brain to a certain level. They need to follow certain steps and certain procedures. Each cell needs to know who the primary partner is. They need to stay with that partner. And some early in disease, when these interactions are altered by factors that cause disease, that's going to change how brain functions and health of the system. In retinal, it's going to affect the vision, ability to see. And in stroke, it's going to affect, depending on the area it happens, it's going to affect the function of that organ that is controlled by that area. But there is a prescriptive power in this knowledge. What can we do about this? Now that we know there are certain elements and playing certain roles in certain places for specific function, now we can act on this. And that's what we're doing about this. So one way, if parasites don't respond that well, maybe it's a decline in neuron signal that drives them. Well, we can boost the neuronal function. That's what we're doing here. So what if parasites can respond to the stimulus well? Well, now we have tools that we can boost their function, or both genetic tools and pharmacological tools. What if they stop talking to each other, as I showed you? The signal, yes, they hear the signal is strong, but they fail to communicate it to supply and branch, and they never hear because they don't drive in here. Well, there are also ways that we're exploring how to repair the way they talk to each other. Just an example. This is kind of complicated, but to give you idea that we are working on this. This is the last slides that are why we actually doing this, because now with that knowledge, we can do something about it. There are tools, optogenetic stimulation. What is optogenetic stimulation? Basically, you can take any cell and using genetic tools, you can make these cells respond stronger. And usually this activity is driven by light. Happen in the retina, it works perfectly because retina is exposed to light. So by putting these optogenetic actuators, it's a channel rhodopsins here, you can actually stimulate neurons, boost the activity that will drive capillaries to dilate. And as you hear, you can basically open them up and allow blood to flow. That's one way of boosting neuronal activity. Well, how about we just bypass the neural activity altogether? We'll just dilate capillaries to begin with on their own. So there are ways to do that. You can use all these genetical tools where you can put specific elements that force these cells to open up the blood capillary. And we're doing that too. And it's a different type of compound. It's a halorhodopsin. The one was chalrhodopsin that activates but constricts. This guy is a hyperpolarized and dilates. And if you put this in a capillary, it actually makes blood flow more. And here it's basically all these red arrows show increase. And all these red arrows are bigger than blue arrows, showing that once you have that actuator, once you dilate the capillaries, it's actually more blood is flowing to the brain. And now we're applying this knowledge to the disease. So now, and a last part is, you can make them locally dilate. You can locally activate neurons, but there will be nothing if they don't signal to each other. Well, this is how you do it here. So in the signaling to each other, as we show, relies on those connecting 43 gap junctions. And we found the ways we can do them both genetically and pharmacologically. We can actually either maintain them, keep them as disease progresses, make it resistant to diabetes, Or if they already lost and they're not coming back, we can genetically actually using these tools like a viral infection and putting insertion of the viral mediated insertion of connexin 43 in specific regions. You don't want to put it everywhere because it's going to disrupt the brain function, but we have specific vehicles that recognize specific cell types and puts this in those cells how it's supposed to be by nature, and only those cells. That's the important part. And by doing that, we can actually able to restore the sense of communication between them. And with that, I would like to thank members of my lab. It's a very strong, hardworking team, and I'm very grateful for their work and effort. Also, my collaborators who provided a lot of insightful discussions and shared resources. Also, numerous funding agencies over the years supported this line of research. And with that, I'll be happy to answer your questions.
Thank you so much, Botir. Excellent, excellent talk. I just want to remind our audience that you can ask questions via the chat window, the icon on the bottom middle of your screen. But I do have some that have come in through other means. One participant who actually, Botir, is thinking about giving up Coke now after listening to your talk, Coca-Cola.
Good idea.
He asked, is early detection part of a regular physical checkup?
Yes.
If not, what would make someone go for a test to find out? Blurry vision, or is there some sort of signal? And if so, what does that look like?
No. So basically, it's very simple. Obviously, you need obviously you need to go to ophthalmologist for this. Optometrists can do this, but they're usually more responsible for corrective errors like glasses, but they do have tools for scan your eye. But the idea is to go to see ophthalmologists because they can do tests called functional hyperemia test. Functional hyperemia test is basically, what is functional hyperemia? It's activity-induced change in blood flow. In the eye, it's accomplished by doing flicker light response. They stimulate with a flicker light and they see how much blood flow changes in your eye. And there is a certain level when they do this light flicker, the blood flow should speed up. And if it doesn't reach that level, that's an early indicator there is something going on. And this happens way ahead of any other irreversible structural changes. So that's the one of the tests that good to do. Also, more simplified stepping back, they do fluorescein angiography. Always they put eye drops with a fluorescent dye and they see how blood vessels are perfused. That's another going to be indication, because if it doesn't perfuse well enough and doesn't fill in well enough, that's kind of indication blood is not flowing as fast as it should be. So there are ways to do. And if you're concerned, I would take it up with the ophthalmologist. Obviously, the primary care physician, refer to ophthalmologist and just go and see what they have to offer. But there are a few things that can be done without waiting.
Excellent. Also, another great question. What can we do to have optimal eye health as we age?
Optimal eye health, obviously, well, physical exercise. That's I'm not gonna, like in my language they say, discover America. Physical exercise, healthy lifestyle. If you have genetic the history of this kind of eye disease, watch for symptoms on that and see how they evolve in your parents or relatives. Eating carrots, vitamin A, obviously. Taking vitamin supplements and reducing computer work, iPads, iPhone, not staring on those. And there is several ways to do. And when you say eye health, it all depends what you mean by eye health. Is it simply avoiding the increase in the prescriptive power lenses, or there is some more serious changes, and that will be talking to your physician, obviously.
Great. And then we have another question. Are there any molecules that stabilize, keep gap junctions open? And do you think they can be used to improve parasite communication and blood flow to prevent or delay diabetic retinopathy?
The answer is yes and yes. So yes to both questions. I cannot say the name because they're not FDA approved for this purpose. We are using for our study. And there are some pharmacologic agents that have been in Europe being tested of all disease for cardiac arrhythmia. Happens to actually the same gap junction connexin 43 is one of the major causes of heart arrhythmia because heart pacemaker cells talk to each other and synchronize their response using the same molecule as the blood vessels communicate. And one of the drugs, candidate drugs, that is now, I think it's a third phase clinical trials, that is actually maintains connexin 43 in those cells. And we have evidence that it works very well in the retina too. But I cannot reveal the name because it's not approved.
Okay.
But I can send later some papers that show, and you make your own judgment.
All right. So you've described in your talk that neurons are smart cells. Vasculature are obviously the blood vessels. Can you go over again about glia cells and what they do?
Well, so now let me see. Let me get that. Yeah. So I'm still sharing screen, right? So now.
Yes.
So the glial cells, actually they outnumber neurons 10 to 1 in the brain. So it's the most popular cell in the brain, not just retina. And for many, many years, it thought they have a supportive role of brain function. They provide essential elements, they metabolize, and they role in conversion of certain substance that neurons cannot consume, but they need, and the glia does it for them. They also the cleanup. I like the title that was written by Jeff Diamond, one of my colleagues, for the Cell paper about glial function, and he wrote a preview in the journal Cell, and he called glia drops the broom, picks up the baby. So there is a growing now. It's undeniable growing and recognized belief that glia is one of the primary and critical element of the brain. So they're not just supportive; they are active. They play a role in the formation of the brain, both neurons and vasculature, and their maintenance, and most importantly, elimination as well. So they have multiple roles, and all of those roles are critical to brain structure, function, and survival.
And that's probably why there are so many of them.
There are many of them, and there are many types of them. Only one type is labeled.
We have another question. Is there a reason why the eye is more vulnerable to diabetes than other organs might be?
Absolutely, there is a reason. Usually diabetes, one of the common things, it's a peripheral neuropathy. What does peripheral means? Peripheral means at the end, tip, fine. And when I showed you, what do you have in the retina the most? You have a lot of capillaries. Those are terminal peripheral blood vessels, and that makes the retina, where is that thing? That makes the retina very, very susceptible to anything that has a word peripheral in it. Here, just look at it. The lion's share of anything here are the capillaries. Those are very peripheral things, and that's why it's very susceptible to any disease like this.
Oh, and we just got a question, which I think is a good one. You were explaining a little bit about how people, when they go and have an eye checkup, what a doctor can look for. And you mentioned this briefly, but can you help our audience a little bit with the difference between an optometrist and an ophthalmologist?
Absolutely. I am neither, so I will limit to, don't take my words as a prescription. But where is it? I will go to the eye. Where is it? Here. So now, usually, optometrists, primary, what we know, they do other things, but usually it's a corrective error, refractive error correction. So glasses, because they limit usually to the optics of the eye, not the brain part of the eye. and usually optics non-invasive part of the eye. That anything that we correct, because when light travels, it has the goal of any image is to focus right on this spot. This is the fovea in the human eye, and the rays sometimes bend too much. They focus before, or they bend too late. They focus beyond, and that causes defocus. And by putting corrective lenses in the middle, that's what they're doing, and that's what your basic how you see better. Number one or number two? Number one, they changing basic refractive error or the band of the lens. That's a correction of astigmatism, because this is not perfect. Sometimes it changes little curvature. It's not symmetric, and that's why some lenses of the same power have a different effect, because they're a little curved. They curved not symmetrically, one more curved here than here, or other way around, and that's what they're doing here. That's optometrist. Anything that is invasive, like surgery, cataract, LASIK, or anything go behind, that's obviously going to be ophthalmologist. That's an MD.
So Botir, thank you so much for sharing your expertise and your time today. And we will just let everyone in the audience know that we appreciate so much their time and energy and participating today. Please share. We're going to be, we've recorded this session and we will be releasing that link shortly. Please share it with anyone in your circle that you think could benefit from some of the subject matter spoken about today. And lastly, we'll be sending out a questionnaire. So if there's any suggestions you have moving forward with this webinar monthly series from the Burke Neurological Institute, we'd be happy to hear it. We want to wish you all to have a great week. Stay well, stay safe, and stay informed. And with that, we'll say goodbye. Thanks again, Botir.
Bye. You're welcome. Happy to be here.